The overall goal of this fluorescent imaging technique is to visualize and quantify cellular proteins in fixed cells at subdiffraction limited resolution using a form of super-resolution microscopy called ground state depletion microscopy and individual molecule return.
This method helps answer key questions in the fields of cell biology and membrane biophysics, as it circumvents diffraction-limited resolution, which helps investigators more precisely define, visualize, and quantify the distribution of cellular proteins.
The main advantage of this protocol is that it enables the acquisition of images with an approximate 10-fold improvement in resolution over conventional fluorescence microscopy imaging approaches, such as confocal microscopy.
Demonstrating the procedure will be Oscar Vivas, a postdoc in my laboratory, and Karen Hannigan, a postdoc in Rose Dickson's laboratory.
To begin, prepare the cover slips and cells as described in the accompanying text protocol. Then fix the samples and perform immunocytochemistry in order to label the plasma membrane and/or intracellular membrane proteins of interest. Next, prepare the oxygen-scavenging Glox solution by first adding 12.5 microliters of a catalase stock solution, 3.5 milligrams of glucose oxidase, and 0.5 microliters of 1 molar tris to 49.5 microliters of PBS in a 1.5 milliliter microcentrifuge tube. Close the tube and briefly vortex it to dissolve the components. Then centrifuge the solution for 3 minutes. Now prepare the photoswitching-inducing thiol solution by dissolving beta-mercaptoethylamine solution into PBS to a concentration of 100 millimolar and modify